Nick Majerison here. Welcome to Top Med Talk. Thanks for downloading the podcast. If you're enjoying what you hear, do make sure you've subscribed. This week we're presenting to you our Xtreme Everest mini-series. Today's presentation is called Epigenetics of Hypoxia. It's Kay Mitchell. Have a listen.
Top Med Talk.
Thank you for giving me the opportunity to come and speak to you today about the epigenetics of hypoxia. And it's difficult to talk about epigenetics without talking about genetics first. So I'd like to give you a bit of a potted history of some of what we know about DNA. In 1843, Mendel described Mendelian inheritance, and he talked about dominance and recessiveness in relation to phenotype. And Levine in 1929 introduced the concept of nucleotides. These are the DNA being made up of strings of nucleotides which contained a base attached to a phosphate group and a sugar. And these nucleotides were either adenine, cytosine, guanine or thymine. And Chargaff in the 1940s further refined this, and he showed that these bases were paired up. So adenine is always paired with thymine and cytosine is always paired with guanine. And then we go to 1953, and we've heard a lot about a particular significant event in 1953 in relation to climbing, and this was when Hillary and Tenzing got to the top of Everest, but something else was going on in relation to DNA as well. And during this year we saw the production of this photograph, Photo 51. And this has been described as the most important photo ever taken. And this is an X-ray diffraction image of DNA taken by Rosalind Franklin. And this photograph supported the announcement by Watson, Crick and Wilkins of the double helix structure of DNA. And these three guys won the Nobel Prize for medicine for this announcement, but Rosalind Franklin was not so honoured. So we know that DNA is double-stranded. You get some DNA from your mum, you get some DNA from your dad. And you can see here this adenine paired with a thymine and the cytosine paired with a guanine, and these are bonded by hydrogen bonds. What else do we know about the human genome? Well, if you stretched out the DNA in a nucleus, it would stretch for about 2 meters, but it's very carefully packed. Most nuclei are less than a micron in diameter. And one of the ways that it does that is you get 147 base pairs, and they're wrapped around these histone molecules in groups of 8, an octamer of 8 histones. And this DNA contains 20,000 genes that code for protein. But those 20,000 genes only make up about 1.5% of the total DNA. So there's about 98.5% of DNA material that we don't know quite what it does. But 99% of our genome is identical across all individuals. So a bit about genetics and hypoxia tolerance. Mike has showed this before. This is the angiotensin converting enzyme gene, and there are 2 alleles, I and D. And in a seed-dwelling population, on the left there's a normal distribution. But on the right, with the elite high-altitude climbers, the distribution of alleles is not normal. And this is the same with critically ill patients. So if you have a D allele, your risk of mortality if you develop acute respiratory distress syndrome is much higher. So there's an association between having at least one copy of the I allele that seems to suggest that you might do better in hypoxia. Hennis and colleagues did a review of genetics in relation to exercise in hypoxia, and they looked at exercise modalities such as running, mountaineering performance and maximum oxygen uptake. 13 genetic polymorphisms were studied, and 4 of these were associated with hypoxic exercise performance, but the I allele of ACE demonstrated the highest association with high altitude performance. And Andrew Murray has showed us these slides before in relation to the work that was done in 2013 comparing Sherpas to lowlanders, and he talked about this allele of PPAR alpha that was enriched in Sherpas, showed that they had adaptations to hypoxia related to oxygen efficiency, reduced capacity for fatty acid oxidation, and improved muscle energetics. So the medical world has become increasingly interested in what genetics can do for us. And in 2000, there was a press conference at the White House, and a UK newspaper commented on this press conference with the headline, "Cracking the Human Code." And this press conference was the announcement that the human genome had been sequenced. President Clinton said, "Today we are learning the language in which God created life." And Lord Sainsbury said, "We now have the possibility of achieving all we had ever hoped for from medicine." But this use of the word code is perhaps a bit misleading. Nessa Carey wrote an excellent introduction to epigenetics in 2011, and she said that DNA is more like a script, it's not a code. So you will have and she made this analogy to Romeo and Juliet, where you have one script by William Shakespeare, but two directors take that script and produce two very different films. And we see this in organisms. So you have a multicellular organism, and all of the cells are genetically identical. They have that same script, but they're phenotypically different. And this is because of this concept of differentiation. So you have a zygote, which is that single cell which contains that DNA from your mum and your dad. And as that cell starts to divide, differentiation starts to take place. First of all, you get that differentiation into the ectoderm and the endoderm, and then further into various different types of cells. And you never get the situation where cells really become less differentiated. You don't get teeth in your eyeballs. And Waddington, who's an embryologist from Edinburgh University and a bit of a polymath, came up with this concept of an epigenetic landscape. And if you think of this ball here as being the zygote, as it starts to divide and rolls down the hill, the cells are differentiating down into very specific cell types, and it becomes very difficult to try and push them back up to become more like stem cells or to get them to take on a different phenotype. And we see this in monozygotic twins as well. They are genetically identical, they have the same script, but they're phenotypically different. And this is illustrated by in relation to schizophrenia. In the normal population in most countries, the incidence of schizophrenia is between 0.5 and 1%, but it's much higher in monozygotic twins. So if one twin develops schizophrenia, the other twin has a 50% chance of developing schizophrenia too. So they've shared the same DNA, they've shared the same womb, and they've probably grown up in a very similar environment. So you would expect that the incidence was much higher. And it may be because of this idea that your phenotype might change through the life course. So again, your genotype is unaltered, but your phenotype may change. And Barker talked about this thrifty phenotype, where he said fetal growth is associated with health in later life. And if we look at this graph on the right-hand side, and I can't see if I can, what you've got along the x-axis at the bottom is birth weight, and what you've got along the y-axis is weight at 1 year. And this is in men who were breastfed, and they were looking for the relative risks of ischemic heart disease. And we've got an arrow here for the average weight at birth and average weight at 1 year. So if you were born small and stayed small, your risk of developing ischemic heart disease was much greater than if you were born normal, or if you were born small but by the age of 1 year had actually recovered and was at a normal weight. And we see this as well in relation to the Dutch hunger famine from World War II. Now, this was an episode where the Allied forces managed to liberate the northern part of Holland, but it took another 9 months or so for them to liberate the southern part of Holland. And the occupying forces in the southern part of Holland stripped the land of food. And so people starved in their thousands. Over 20,000 people died. And the Dutch government looked at people for generations afterwards who were exposed to that famine. So in particular, they looked at the babies that were born around that time. And the babies that were conceived before the famine, but were born during the famine, were born small and stayed small. Those babies that were conceived during the famine and born after the famine were born normal, but they had a much greater risk of obesity. And this risk of obesity was seen in subsequent generations. And the reason I have a picture of Audrey Hepburn here is that she was actually lived during that period of famine. She was 16 years old at the end of it, and the after effects of living through that period of time for her were poor physical health that remained with her for the rest of her life. So what I'm talking about here is epigenetics. And epigenetics is the study of changes that affect the transcription and translation of DNA, but not its underlying sequence. And these changes can be heritable and they can be reversible. And the epigenome is deranged in about 20% of cancers. So what we're talking about is the mechanism by which genes are switched on or switched off, transcription and translation. So you get a transcription factor which attaches to a promoter region in a gene, and that enables an RNA copy to be made of the gene, and then that RNA copy is translated into a protein. And if we look at this in relation to hypoxia, we've talked already about HIF1, which is responsible for maintenance of oxygen homeostasis and vascularization. HIF is a transcription factor, and it attaches to a binding site, a hypoxia response element. And depending on which genes that HIF attaches to, it produces different proteins. So there's erythropoietin, there's nitric oxide synthase, there's VEGF, and homoxygene 1. These are all gene products that HIF1 can activate. So how are these genes switched on or switched off? Well, we talk about these epigenetic mechanisms, and the two most common of those are DNA methylation, and this is where a methyl group is attached to a cytosine, which is followed by a guanine, so a CPG site. Now, this isn't a CPG site with a hydrogen bond between the two strands of DNA. This is on one single strand of DNA where you have a cytosine followed by a guanine. And then you also have histone modification. And you remember I talked about those 147 base pairs wrapped around the octamer. They have tails. And if they're acetylated, then that is an epigenetic mark. So if you have your DNA that is not methylated, and your histone tails are acetylated, the DNA is much less compacted and the gene is switched on. But if your DNA is methylated and you have no acetylation of your histone tails, then your DNA is much more compacted and transcription doesn't take place. So this is a very new area, and there are not an awful lot of studies out there, but I'm going to show you a couple of studies that have looked at epigenetic marks in relation to hypoxia. The first is a study looking at the hypoxic regulation of epigenetic modifications during disease progression in rheumatoid arthritis. Now, you get synovial fibroblasts, which are a key player in this disease pathogenesis, and hypoxia is thought to be a possible culprit for this aggressive phenotype. Your inflamed joints are hypoxic, and Heron and colleagues were looking at H3K27ME3, which is a histone that shuts down transcription. And they had two groups. So on the left-hand side, they had some synovial fibroblasts, which they cultured 50% of them in hypoxia and 50% of them in normoxia for 24 hours. And then they also took some human synovial tissue from 3 different groups. So we had patients who had developed rheumatoid arthritis symptoms for less than 3 months. We had patients who developed non-rheumatoid arthritis symptoms for less than 3 months. And then we had patients who developed rheumatoid arthritis for more than 3 months. And in the cultures, they showed that H3K27 had significantly increased in fibroblasts exposed to hypoxia compared to those that were in normoxia, and that this was associated with HIF1 alpha induction. And in those cells from the patients, GLUT1 and H3K27 were increased in early rheumatoid arthritis patients, but not in the other 2 groups. And they concluded that the changes in transcription and translation occurred early in the onset of rheumatoid arthritis and were associated with hypoxia. But these changes were not apparent later in the disease progression, suggesting that there had been some adaptation to chronic hypoxia. And the second study was from Julian and colleagues, and they were looking at epigenetic modification of gene expression during human acclimatization of hyperbaric hypoxia. And they took PBMCs from 21 healthy individuals at 4 time points up to 5,260 meters. And they carried out 450K in Affymetrix arrays. And after 16 days, they saw 183 different sites on the DNA demonstrated changes in levels of methylation. And these were associated with genes such as EIF2C and heat shock protein, which are associated with hypoxia. And they carried out a methylated quantitative trail locus analysis, which identified altered transcriptional activity in genes considered central to human acclimatization to hypoxia. So their conclusions were that methylation status altered the transcription of the genes that was associated with acclimatization to high-altitude hypoxia, and that these changes had been seen within 3 weeks of exposure. So just finally, what is the clinical relevance? Well, we saw 2 studies there. One was a study where we looked at methylation changes between different groups of patients, and the other one was where we looked at one group and we looked at how their methylation changed over time. And you can collect samples of DNA and look at these epigenetic changes and see what genes that they are related to. And there are public databases that are collecting from the research that's being done every single day. They're collecting information about the annotations for all of those genes. So when you do your study, you can look at which genes have got particular differences in methylation, and some of those will relate to genes that we know about, like EPO, that we expect to see with the phenotype that we're investigating. But some of those methylation changes may be on genes that we're not expecting to see, and it may be that that information gives us additional information about what to target when we're looking at changes in phenotype. So then you refine the model and you go back again. So we're going to continue to do this, doing studies in patients and doing studies in chambers. So it will be a question of watching this space. Thank you.
Don't forget you can meet the TopMed Talk team, ebpom.org/meetings. Our next big event takes place between the 28th and the 30th of September. EBPOM USA Chicago Masters Course, a perioperative care practicum.